Hi everyone, in this section we will talk about gene engineering. After watching this video, you should be able to tell the workflow of genetic engineering. Gene engineering, also called genetic engineering. Nowadays, it is a widely used technology in modern biology. We know that recombinant DNA is DNA with a new nucleotide sequence. Such DNA is formed by joining fragments from two or more different sources. In this section, we will focus on the workflow of genetic engineering. The basic steps of genetic engineering can be briefly described as shown in this figure. Firstly, isolating DNA to be cloned. Secondly, use a restriction enzyme or PCR to generate fragments of DNA. And thirdly, generating a recombinant molecule by inserting DNA fragments into a vector. And fourthly, introducing recombinant molecule into a new host. Finally, screening the successfully transformed host. Generally, they will exhibit a new trait due to the expression of the transformed gene. To obtain target gene of interest is the first key step for genetic engineering. The target gene can be extracted from microbe, animal, or plant. It can also be synthesized of cDNA from mRNA via reverse transcriptase or chemically synthesized. The next step is selecting the suitable genetic vector for transformation. There are several requirements while choosing the vector. Firstly, it should be a replicator with low molecular weight. It can replicate. Secondly, it can multiply in receptor cells to a large number. Thirdly, it usually have only one restrictive endoenzyme site so that the target gene can be integrated into the vector DNA. Fourthly, there must be a genetic marker for efficient screening. For example, some vectors have antibiotics resistance site so that they can survive after growing on medium with antibiotics. The next step is integrating the target gene with the genetic vector. The DNA sequence of both target gene and the genetic vector can be catalyzed by endonuclease into sequence with two complementary cohesive ends. Look at this picture. It shows the sticky ends in the vector cut by endonuclease. By mixing the processed genetic vector and the target gene, a recombinant vector will be formed under the catalyzation of ligase. Then, the next step is introducing the newly formed recombinant vector into receptor cell, wherein the target gene would be expressed. Then, after screening and identification, the newly transformed strain with desirable trait could be obtained. Genetic engineering has been successfully used in microbes, plants, and animals, which will be helpful for sustainable development. Finally, let's take a look at the keywords in this section. Okay, that's all for this section.